am Madam Joyce from Educare TZ. I'm teaching biology, and today I will be with you to discuss our topic, which is found in class level Form 5. And here we are going to discuss the cytology, whereby we are going to discuss, or we are going to deal with our subtopic, which is the biochemistry. Whereby in our subtopic, which is biochemistry, we are discussing on the carbohydrate, specifically on the class which is the disaccharide. So in the previous session, we tried to see in the properties, or we terminate by looking on the properties of the disaccharides. We have seen there is sweetness in taste, they are non-reducing sugar and reducing sugar, they can be hydrolyzed, and so many properties. So my student, you are welcome in this session, whereby we are going to discuss the structure now, or we are going to discuss the formation of different kinds of disaccharides. And here we are going to start with the first disaccharide, that is the maltose, or it can be termed as the multi-sugar. So we are going to start with the maltose. So what is this now? The maltose is a double sugar which occurs naturally in roots and the radicals of germinating cells such as maize, sorghum and finger millet. So tunasema kwamba maltose ni multi-sugar, yani sukari ziko zaidi ya mbili, so that is multi. So this multi-sugar tunasema kwamba they are double sugar which occurs naturally in the roots and the radicals of germinating cells such as maize, sorghum and the finger millet. So in their root structure, or in these structures, or in this part is whereby you can find the maltose. Kwa maana kwamba ukienda kwenye mahindi together with the sorghum and the finger millet is whereby you can find the location of these maltose. So now we can say that artificially it is made up by chemical combination of two alpha glucose units. During this combination, hydroxyl group at carbon number 1 and carbon number 4 of these glucose residues are involved in the formation of oxygen covalent bond called glycosidic bond. Hapa tumesema kwamba naturally hii maltose inaokaa kwenye these parts, the roots of radicals of germinating cells such as maize, sorghum and finger millet. That is naturally form of this maltose. But artificially, this maltose can be made. And how is it made? Here we have seen that it is made by a combination of alpha molecules, which is monosaccharide, whereby inatengenezwa vipi sasa is due to the combination of hydroxyl group at carbon number 1 and number 4 of two glucose residues. And now they are involved in the formation of a covalent bond, and that covalent bond, it is termed as the glycosidic bond. So now let's see, that is, since it is formed between carbon number 1 and carbon number 4, then it is termed as the 1,4 glycosidic bond. So this process involves condensation reaction, thereby therefore a molecule of water is lost. We're going to see all of the analysis, all of the explanation in the figure 1.21. Tumesema kwamba hii structure, in order for the formation of this maltose, there must be a presence of two molecules of glucose. Tukiwa na two molecules of glucose, ndipo ambapo sasa we can artificially made the maltose. So how? Let's see this figure. This is a chemical combination of two alpha units to form maltose. So we have the first unity of alpha glucose, and also we have the second unity of alpha glucose. Why is it termed as alpha? Kwa sababu the hydroxyl group it tend to point on the below of the ring. on the carbon number 1. You can see in this carbon number 1 and this carbon number 1. So now the hydroxyl group from alpha carbon of this one, it tend to combine with the hydrogen group of the last or of the next glucose, ndipo ambapo sasa ita result into condensation. So here two molecules of or two atoms of hydrogen tend to combine with the oxygen, ndipo ambapo inapelekea kupotea kwa maji, na hiyo ina bring about condensation reaction. So the result of condensation reaction is the result of formation of glycosidic bond. The glycosidic bond, it is formed between carbon number 1 of the carbon atom and also carbon number 4 of the other glucose. Ndio ambayo sasa ina bring about the formation of this maltose. And remember, the bond which is formed between the bond which is formed between the glucose and another glucose of alpha, that is bring about 1,4 glycosidic bond. Why is it termed as 1,4? Now you can see here, this one is carbon number 1, but this one is carbon number 4. Start counting from here. That is 1, 2, 3, and this one is 4. Ndio ambayo ina bring about 1,4 glycosidic bond. So that is how the maltose it is formed. Hivyo ndivyo maltose inavyotengenezwa. It's just a combination of two molecules of alpha glucose, thereby it results into condensation reaction. That is, a molecule of water, it tends to be removed, and thereby it tends to form 1,4 glycosidic bond. So maltose is a reducing sugar since it has free aldehyde group in its molecule. So tunasema maltose hii ni reducing sugar. Why is it reducing sugar? Because there is a presence of aldehyde group in its molecule. So the presence of aldehyde group in its molecule ndio ambayo inapelekea sasa, if there is donation, it tends to donate a pair of electrons into another molecule, and thus bring about reducing nature in maltose. And in one of the glucose units, the aldehyde at carbon number 1 has been used in the formation of the carbon, of the bond, which is the glycosidic bond. You can see in this figure, that is in the carbon number 1. In the carbon number 1, this one is the aldehyde group. So one of its atom ndiyo ambayo sasa imetumika kwenda kutengeneza ile glycosidic bond, or that bond formation. And while the second glucose unit, ile unit nyingine sasa ya glucose, tunasema kwamba the aldehyde at carbon number 1 remain intact. Since the carbon involved in the formation of the bond is that at position number 4. Kwa hiyo sasa ile carbon nyingine au ile hydroxyl nyingine au ile aldehyde nyingine inayopatikana kwenye ile structure now ya glucose ina remain intact, kwa maana kwamba ina remain the same, haiwezi katumika, ambayo sasa ile iliyobakia kwenye carbon number 1. Lakini ile ambayo inapatikana kwenye position number 4 ndio ambayo sasa inatumika kwenda kutengeneza the hydrogen bond. That is this one. This is structure. Now you can see in the carbon number 1, the aldehyde group remain intact. But in the carbon number 2, or in the glucose number 2, that is in the carbon number 4, ndipo ambapo sasa ile hydroxyl group inapatikana kwenye ile aldehyde group ina donate kwenda ku lose the water in the structure. So that's all about that's the explanation. And now that's all about the maltose. So here the thing that you have to remember, maltose inatengenezwa na kitu gani? Usiseme tu glucose plus glucose. You have to state the presence of alpha glucose and alpha glucose. So two molecules of alpha glucose ndizo ambazo zina result into the formation of maltose group. or maltose, that is disaccharide. So now let's see another disaccharide, that is sucrose, which is cane sugar, or you can simply say the sugar cane. So hii sucrose inaitwa cane sugar, sugar cane ni ile sukari ambayo inayotokea kwenye miwa. So ile sukari tunasema kwamba inaitwa sucrose. And this sucrose, we have seen that it is mostly found in all plants. And now here you can see this sucrose, it is a double sugar that naturally occur in the stems of sugar cane plants. So hii ni double sugar ambayo sasa naturally inapatikana kwenye sugar cane. Lakini sasa hii sucrose artificial tunasema kwamba it is made up by chemical combination of galactose, that is aldose sugar, and fructose sugar, that is alpha ketone sugar. So tunasema simply sucrose inatengenezwa na galactose together with the fructose, and that fructose, it is the alpha fructose, ndio ambayo inahusika kutengeneza sasa hii sucrose. So my student, for more clarification on how this sucrose it is formed, I would like to invite you in the next part. We're going to clarify in detail how is it formed and its structure in a general. So thank you for your attention and for listening.